Welcome, everyone. Today we will have 5 speakers, and our first speaker to start this important session is Dr. Trudy Paap. She's a research fellow in the Forestry and Agricultural Biotechnology Institute of University of Pretoria in South Africa. She works as a forest pathologist, and she deals with a lot of plant health issues, not only in South Africa, but also from Australia. Her research is focused on the impact on native and invasive pests and pathogens of natural and planted forests and urban trees, and she has a lot of experience in surveillance and detection of new and emerging pests. So Trudy, please share the screen with us.
Okay. Well, thanks heaps for the invitation to speak today. So I'm going to talk about a project that we've been running in South Africa's botanical gardens for quite a few years now. But okay, so it's pretty clear that forests globally are increasingly under threat from invasive pests and pathogens. And this is largely a consequence of international trade, and that's something that's not going away. And a few major pathways have been recognised as the primary means by which invasive pests and pathogens are moving around, and that's through wood and wood packaging materials and the movement of plants and plant material. So one of the systems that's been established to try and deal with this is the use of sentinel plants. This is from a point as well that a lot of these insects and microbes that become problematic were either not known to science prior to them arriving somewhere else, or they're not damaging in their natural environments because they've co-evolved with their plant hosts in those environments. And so by looking at non-native plants in other countries, we can look for native insects or microbes that could potentially cause damage on those plants. And we can also use sentinels in terms of early detection. So these would be plants that are in high-risk sites, so urban areas or sites near harbours, ports. And botanical gardens are key sentinel sites. There's thousands of botanical gardens worldwide. They house a huge number of known plant species, and you've got dedicated people working in those collections on a daily basis. So they really function quite well as sentinel sites. Some years back, under BGCI, the International Plant Sentinel Network was established, and this is a platform to coordinate and exchange information and support sentinel plant research within botanical gardens and arboreta, with its objectives being to function as early warning systems to identify new and emerging pest and pathogen risks. So back in 2016, a project was established in South Africa, and it's a collaboration between Forestry and Agricultural Biotechnology Institute at University of Pretoria and South Africa's National Biodiversity Institute, or SANBI. And the aim of the project is to use the IPSN as background to improve surveillance and identification of new and emerging pests and pathogen risks in the country. I'm originally from Australia, but I was lucky enough to be able to get the first postdoc post in that role. The project looks at gardens and some arboreta across the country. It's an amazing country with many different biomes, many different climate types, so incredible collections of plants to work in. I was a postdoc for the first couple of years, and since moving out of that postdoc role, we've had 2 others in that role, Ms. Ngosa and Felipe Voloki, who's the current postdoc. And it's a project that I've remained involved in throughout my time here in South Africa. And the project has given us lots of research opportunities to inform plant health. We've been able to increase our understanding of the impacts of known pests and pathogens. We've identified new pest-host associations, and the project has really worked as an early warning system as well. A lot of things have come out of the project, but in terms of first reports and first records on new hosts, there's several publications that have come out. I've just highlighted two interesting ones of note here. But because of the way that botanical gardens operate, all the people coming into them, you get travellers arriving from other countries that will get off the plane and go straight into a garden. There's also a lot of movement of plants and plant material and organic matter. So botanical gardens can really be hotspots for the introduction and spread of pests and pathogens. So it's really vital that the staff working in those collections consider biosecurity as an essential consideration for all the activities that they're doing there. One of the benefits that we've had from running this project for all these years now is that there's been real opportunities for capacity building. The level of capacity across the gardens is really varied. Like some of the collections at the moment don't even have a qualified horticulturalist working there. So there's real resource constraints that you're dealing with as well, but lots of opportunities for engagement. Some of the things that we've done is like running monitoring and best practice training workshops. Whenever we go to the gardens to do collections, we work with the staff there, and we've really had a lot of support from groups like the IPSN, and we've had other international experts come and join us with some of these workshops as well, which has just been fantastic. So we've run training workshops, which include seminars and discussions. We've had pruning demonstrations, walk through the gardens to identify existing issues in the gardens, setting up things like footpaths at entry points for their greenhouses, and where issues exist like Phytophthora dieback, setting up treatments. Like here you can see phosphite injections taking place. And the culmination of all of that work we've put together in a best practice document. This is something that's been developed between us at FABI, and then the Horticultural Enrichment Forum of SAMBI. And the HEF is a group that's comprised of some of the senior horts in SAMBI. And this is something that started several years ago. It was really Pendulo, my co-editor in this document, that has driven getting things to this point. So we've had quite a few contributors to the document, including the other FABI postdocs and several HEF members. It's been an amazing process to be part of. So it's something that started years ago just as an idea, and it's taken a long time to get it into a solid document. It's in layout at the moment, and it will be published. SAMBI has something called Biodiversity series, so hopefully that's not too far away. Unfortunately, because it's not being fully produced yet, I can't show you the nice graphics on the inside, but I will just walk you through a couple of the things that we've done. So we have gone through and identified the major threats and the pathways of pest movement, and then described these threats, given recommendations to address it, and then we've got other links sort of within the document that give more details. As an example, we've created a flowchart and decision tree for a quarantine procedure for all incoming plant material, everything from sort of giving a threat or risk level to whatever material is coming in, setting up record keeping systems, and then how to deal with the material depending on the threat level that has been identified, and what to do when issues arrive. Because we've got people working in these collections every day, and because South Africa is a really big country, it's not possible for the current postdoc to be in those gardens constantly looking for issues. But because we've had chances to really help get the staff's eye in for when there's issues, we can have them looking out for emerging pest and pathogen issues. And when they see that something is going wrong, rather than just ripping out the plant and throwing it on a waste heap, they can go through a process to sort of identify whether it's something that should be sent to us. They can also just send us photos, so we give instructions on how to take photos. And then if they want to submit a sample, we've set up a system for sample submission. And because we have so many people coming through these gardens, there's also a real opportunity to engage with visitors to the garden, to educate them as well about pest and pathogen issues and why hygiene and biosecurity best practice is important. So we've identified some of the key messages that the gardens can be communicating to visitors through interpretation, signage. So I want to wrap it up here, but I also just want to acknowledge that aside from the people in Sambi, the gardens themselves, and the other postdocs that have been involved in this project, we've had a lot of support from different institutes across the world. Like I mentioned, we've had a couple of wonderful international experts like Brett Summerall, Chris Malafy join for some of these training workshops and give their input into the things that we've been developing. John Wilson and Mike Wingfield have also been really key people in helping drive this project and to make sure that it has the life that it has. And then Lara Salido from the IPSM has just been wonderfully supportive and just always fantastic to engage with. So thank you all for listening.
Thank you, Trudy, for an amazing presentation. Do we have any questions? Let me give one moment. Okay, so in the chat, we have one question from Josephine. Her question is, how transferable is the biosecurity document to other parts of the world?
I would say very. So one of the really wonderful things about working with the group that we worked with is that they know the limitations that they're dealing with in terms of finances and resources and what's accessible and what's not. And if it was just a bunch of plant health experts sitting there going, well, this is what we think should be done, then it probably wouldn't be as practical and as usable a document, because we want it to be something that is usable. And I really think that that's one of the key things about it, is that it is practical and it's just giving simple solutions, really basic things like when you're moving between beds, clean your tools and clean your boots, nurseries, getting plants up off the ground. And if you haven't got the possibility to set up really fancy infrastructure like benches, just having some wire mesh on bricks, keeping weeds out, just really, really simple, practical things. Okay,
so there's a follow-up question. How do you determine the threat levels?
So there's already quite a bit of literature around that, and it's firstly where the plant material is coming from. So is it coming from overseas? Is it coming from another part of the country, like a different biome? The size of the material, is it in soil? Is it bare rooted? So things like tissue culture provide or present a much lower risk than, say, a large potted plant.
And I would like to ask you something additional. So what would be like the first recommendation that you do to this botanical gardens do you do also recommendations for, let's say, they plant a specific plants that they can always use as a sentinel plant or as a primary source, that usually is like the ones that you say that these ones are getting infected most of the time?
We haven't really made those sorts of recommendations. We tend to use what's already present there as a way of diagnosing potential issues. I mean, we do know that there are certain plants that are, say, highly susceptible to Phytophthora cinnamomum. So if you see that particular plant dying, that can be a fairly good indication that that's the problem that you're dealing with. But no, in terms of making suggestions on what to plant, we haven't really gone that route.
Okay. Thank you so much, Trudy. We will continue with our next speaker. Our next speaker is Dr. Jane Stewart. She is an associate professor of plant pathology in the Department of Agricultural Biology at Colorado State University in Fort Collins in the USA. She works on forest, shade, and fruit trees, and her experience is on fungal biology, population genetics and genomics, molecular diagnostics, and management of different plant pathogens. And these also include invasive and emerging pathogens. So, Dr. Jane, we are looking forward to your presentation.
Thank you so much. And thank you to the organizers for inviting me to come talk today. So what I want to talk about is how we can harness the microbiome to enhance forest pathology research. And what I'm going to do is highlight 3 projects that come from my lab from 3 awesome women scientists. So studying forest health in the 21st century is really difficult because we have increasing complexity with emerging diseases as we have increased globalization, climate change, and factors that we hadn't had historically. And so new technologies are really needed to help us develop novel management practices to help these multifaceted issues. So one of those new technologies is a microbiome technology. And I think this is a technology that could revolutionize research and understanding these forest health issues, but then also aid in ecosystem management of these issues. So changes in the microbiome with disease. So a lot of us have heard that the microbiome changes with as disease develops. And a term that was coined really by the human health research arena at the beginning was dysbiosis. And this is basically describes an imbalance that you get with microbial communities once a disease state has started. So here you can look at this tree that I have. And on the left side, we have a healthy tree and we've got a nice balanced microbiome. But once we start to get a pathogen in that root system that is denoted by the yellow star, what we see is that the community shifts and it shifts towards a pathobiome. And a pathobiome describes basically community members of the that are basically associated with disease, and they help the pathogen infect that host. And so historically, if we think about the disease triangle, we thought about maybe one pathogen and one host, but now we know that it's a consortium of organisms. So recognizing these different genetic or underlying factors that drive this host pathogen and microbe interaction may help us basically provide new approaches for management of these diseases. So I'm going to talk about 3 women who come from my lab that have worked within the microbiome space to answer pretty diverse questions. So I just wanted to highlight the different types of questions we can start to answer with microbiomes. So the first project is focused on what taxa are causing the disease. Is disease caused by one or multiple organisms? The second project is, are pathogens endophytes as well? And how is the pathogen attacking its host? And how is the host responding to that attack? And then the third one is looking at how abiotic disturbances change the microbiome and how do pathogens respond. Now, I don't have a lot of time to go into a lot of detail for each of these projects, but I put the papers associated with each of the projects down below so you guys can look at those. So the first work was done by Hope Raymond, who was actually, when she was in my lab, she was an undergraduate, and she's now a managing editor for Carbon Markets. What she looked at was a bacterial pathogen, Lonsdaleia coarcosina, that we have discovered in the US causing 2 diseases. One was drippy nut in California and drippy blight in Colorado. And we know that bacterial pathogens have been increasing over the last 20 years, especially on oak species, and have really increased on a global scale. One thing that we were interested in looking at was, is there a pathobiome associated with Lonsdaleia coarcosina? And many people wanted us to check, are there fungi involved? Is this really a bacterial disease, or are there other fungal organisms involved in the disease development. So basically what we did was we took samples from healthy and diseased oaks from California for drippy nut and Colorado for drippy blight. And then we sequenced the bacterial and fungal communities. And what I'm just highlighting here is this is a PCOA plot. So the further away the dots are, the more significantly different those communities are. And the things that I've highlighted here, so here we have blue dots and we've got orange dots. And the blue dots are asymptomatic samples from Colorado, whereas the orange dots are symptomatic samples from Colorado. And what you see is a really nice separation. So there's a dysbiosis of that community once disease develops. However, when we look at our fungal communities, what we can see is that we've got dots here that are a mixture of green and yellow. And then we have another group that the dots are a mix of orange and blue. And what we see is there's separation not by disease state, but rather by geography. And so we don't see a dysbiosis in the fungal community. And then we can start to look at who is present in that community. So this is looking at relative abundance of the bacteria and of the fungal taxa in California and in Colorado. And here we have asymptomatic samples and symptomatic samples across. So the first thing that I've highlighted here in red is Lonsdaleia. So what we found is that Lonsdaleia is actually present in both asymptomatic and symptomatic samples, both in California and in Colorado, but it does increase significantly with disease. Then we also wanted to look at, are there other bacterial members that might be associated with the pathobiome? And what we have found is that Zymobacter is a genus that is present both in California and Colorado, again, in asymptomatic and symptomatic, but it also increases with disease. And then what we wanted to do was look at the fungi. And what you can see is that it's a very colorful graph with many different taxa, and none of those taxa were associated with basically asymptomatic or symptomatic. And so we really didn't see an association of fungi associated with drippy blight or drippy nut. The next project was done by a PhD student in my lab who's now Associate Professor Jessa P. Atta, and she's a professor at the University of Philippines in Los Baños. And she looked at needle pathogen system on Pinus contorta or lodgepole pine. And so there are two different pathogens that actually infect lodgepole. One is Lophodromella concolor, and the other is Lophodromella montevaga. And what we were interested in looking at was basically characterizing the fungal composition. And then we also did metatranscriptomics, where we were able to then look at the expression profiles of these pathogens in asymptomatic and symptomatic needles, but then also look at the host response to those two infection states. So the first thing to note is that, again, we have a relative proportion of taxa here, and we've got one for Lophodromella concolor and one for Lophodromella montevaga. And what you can see is that in the asymptomatic state for both pathogens, we actually have reads that correspond to both pathogens. And that was something that was really interesting to us. So we've got these pathogens both present in the asymptomatic needles, and it tells us that we may have one that has a pathogen that has a latent or endophytic life stage. However, when we move to the disease state for each of those pathogens, we get dysbiosis that happens. So either Lophodromella concolor or Lophodromella montevaga basically take over the community. So we also wanted to look at whether or not what kind of genes are upregulated when we have infection or asymptomatic needles. And so here we've got, these are differentially expressed genes that are across different protein families involved in pathogenicity. And what you can see here is that there are basically two bars present across these groups, and these represent Lophodromella montevaga is the light blue, and then in symptomatic samples, and then Lophodromella concolor is symptomatic in the gray bars. And so what we see is there's differential expression in pathogenic disease genes in the symptomatic samples, but none in the asymptomatic samples. So they're not producing, the asymptomatic samples are not producing any genes related to pathogenicity. So then we wanted to know, okay, how does the host respond to this. And what we found is that in asymptomatic and symptomatic needles, the host is actually producing plant defense genes that are actually turned on. And so what you can see from this is that this long bar, the dark green bar, is basically samples that are associated with Montavaga, but asymptomatically. So even though the pathogen isn't turning on pathogenicity factors, the plant is actually producing plant defense genes in response to that. And so that's a really interesting understanding of how this latent pathogen is basically interacting with its host. Okay, and so the last project is basically what happens when we add an abiotic factor to this. So how does fire alter the soil microbiome communities in forests with armillaria root pathogens? And this was work that was done by Ada Fitzaxon, and when she was a master's student in my lab, and she's now stayed for her PhD. So basically, fire can be really detrimental, of course, for microbial symbionts, right? So this is a picture that comes from one of our field sites where there was a high severity burn. And so all of the older species, the trees have died off and basically have fireweed down below. And you can imagine those communities are going to change from basically the heat-induced mortality. There's going to be soil changes that happen in the nutrients and the profile of that soil, but also the vegetation community changes. As we lose those overstory trees, we're going to lose communities, microbes that are associated with those trees. And so what we did was we went into fire sites 15 months post-fire, and we wanted to make sure that there was armillaria present on that site. So we used basically sampling methods of armillaria using rhizomorphs or mushrooms. And then we sampled trees that were in 3 different categories: a high severity fire, a low severity fire, and then an unburned site. And so the first thing we wanted to look at was, okay, just how do the communities differ when we're looking at fire influence? And this is using a MNDS plot. And basically, the further away the circles are, the more significantly different they are. And what you can see is that for fungi and bacteria, the high severity burn is significantly different than the low and the unburned, and the same for the bacterial community. So there was change within those communities. And then we also wanted to look at what taxa were basically significantly more abundant in a community when compared to the other ones. And so here we've got a Venn diagram. This is communities that are associated with the high severity burn. We've got the low severity burn in green, and then the communities associated with the unburned in blue. And what we found is actually very interesting. Armillaria solidipes, which is the main driver of armillaria root rot in our area, was associated only with high severity burns. And this could be really problematic. One, because those trees are going to be more stressed in that area, so it's a lot of food source for armillaria solidipes. But also, after a high severity burn, we will go in and plant trees. And so this is a possible inoculum source for regenerating seedlings and for trees nearby. However, when we looked at the unburned and the low, what we found was a high proportion of ectomycorrhizae that sort of disappear in the high severity burn. And one of these, which was really interesting, was Swillis, and it was found relatively more abundant in the low burn. And Swillis is a ectomycorrhizal fungus that basically is effective post-fire colonizer, so it can actually come into sites pretty early on through the movement of basidiospores into low severity burns. And so this kind of begs the question, could we use this as a management treatment for armillaria root disease? So just in summary, I think I just wanted to highlight that the microbiome is a really powerful and versatile tool. As we know, the disease triangle is way more complex than just one host and one pathogen. And this is sort of described here, looking at the microbial communities that are just as important to disease development as our changing climates, host predisposition, human activities, and globalization. And using these different types of tools may lead to novel and important management strategies. And with that, I'd like to thank our funding, and then also members of the Stewart Lab, of course, Hope, Jessa, and Ada as well for doing awesome research. And thank you, and I'll take some questions.
Thank you so much, Ian, for your presentation. There's a question in the chat. The question is from Kadiatou Schiffer. The question is, what could be the drivers of the increase in bacterial diseases on a global scale? Rapidly changing climate conditions or more detection thanks to microbiome tools?
I think it's probably a combination of both, actually. What we found is that the bacterial diseases really like drought-stressed trees in our some of the studies that we've done with drippy blight, but then we are able to detect things better now, for sure. But in Colorado, we have trees that were 30 years old, 40 years old that are just now starting to die off from drippy blight. And so they didn't have the disease before, and it is prevalent in asymptomatic trees. So it may be that there's a change in the host that's causing this disease to develop.
Okay. I have another question. So in case of pathogens that have like a life cycle that can be present in asymptomatic and symptomatic. So those, like the example that you were showing of Lophodermella, are there other pathogens that behave similarly or that you have found on the microbiome that behave that similarly?
Yeah, there's a lot of endophytic, and actually like Diplodia can also do that as well, be a latent pathogen or an endophyte. So there's several different types of organisms that will do that for sure. Yeah.
Thank you so much.
Thank you.
Our next speaker is Tatiana Rojas. She is an economist from Universidad de los Andes in Colombia with a Master in Science in Development Economics and Policy from the University of Manchester. And currently, she is a PhD student at the University of Guggenheim for Tropical Agricultural Science. And she implements research and investment projects in biocommerce and the bioeconomy. She specializes in the development of non-timber products across various rural regions in Colombia. So with that, I will give the floor to Tatiana. So please share your presentation.
Good afternoon, everybody. First of all, I wanted to thank you for this invitation, and let me know if you can hear me well and if you can see also my screen.
Yes, we can see your screen and we can hear you.
Perfect, amazing. So as Gina stated, my name is Tatiana Rojas. I come from Colombia. Right now I'm working at the University of Hohenheim in Germany. I'm a doctoral candidate, and today I would like to present you a glimpse of my PhD research about the biodiversity-based value chains and development pathways for natural ingredients in my country, taking the case of Corozo palm, Bactris guineensis. You can see the palm here in the left side of your screens. I would like to start letting you know that overall, non-timber forest products have been recently taken into bioeconomy strategies, particularly in countries with large forested areas such as Colombia. And this is very important because I take into account a new discourse of the bioeconomy, that is the socio-biodiversity bioeconomy, which in the end reflects and strengthens the recognition of local populations who directly depend on livelihoods and biodiversity use and conservation. But also because this new approach of the bioeconomy embraces the diversity of practices, knowledge, and innovations that not only ensure the ecological sustainability of biodiversity use, but also its cultural aspects. So in this regard, I want you to look at this picture. This is the main part of the plant use of Bactris guineensis, the fruits. So Bactris guineensis comes from the family Aracaceae, which are palms. And according to diverse ethnobotanists in my country, Corozo or Uva elata is considered the most important fruit yielding palm in the Caribbean region of Colombia. But I also wanted to let you know that this palm is really interesting because only 5 countries in the world have its distribution. but Colombia, according to GBIF, has the highest number of biological records. Here also in the right part of the picture, you can see that an individuum can have a height of approximately 4 meters, and that it's an endemic palm of the tropical dry forest ecosystem, which is a very important ecosystem in the tropical world. And in Colombia more, because only 8% of its original cover remains. So it is very important to tackle efforts that can help restoring this ecosystem. And Bactris guineensis is one of the pioneer species that can help us tackling that. Why? Because it grows in clay soils with a high incidence of light, so therefore is very resistant to climate change. And also because it's a fast-growing species with a high productivity. Some studies have revealed that per plant per year, it can produce about 30 kilograms, which is a higher productivity when you compare to acai production in the Colombian Amazon of wild systems, which runs about 12 to 13 kilograms per plant. But more than this, I also wanted to let you know that this palm provides a diversity of contributions, not only to people, but also to living beings, and that is also really highly culturally appreciated in the Caribbean region of the country. To mention some of the material and non-material contributions, in material contributions in Colombia, we have a diversity of uses that end up in different applications that are concentrated in the food and beverages sector, and some of them a bit in the cosmetic sector. In non-material, we have other diverse intangible contributions. For example, the stem is used to produce the guacharaca instrument of the traditional vallenato folk music, which in the end represents the culture of an identity of the Caribbean region. But also from the stems and from the leaves, we also see traditional construction. And overall, this palm also has diverse ecosystem services. It's an umbrella species, that means it's the habitat and the oasis of many other species. It promotes or diminishes erosion, regulates the climate, and also contributes to water provision and regulation as it's located nearby water bodies such as swamps. According to a study that was published in 2022, this palm is also a multi-purpose palm in the bioeconomy. What does it mean? Basically, it means that from the fruits, the seeds, the leaves, and the stem, that these are non-timber forest products, we can have many products and applications in many sectors. However, as I mentioned, in Colombia right now, we're concentrated our applications in the food and beverages sector, but there is a connection and a potential in other 7 bioeconomy sectors, such as cosmeceutics, nutraceutics, functional foods, biofuels, phytomedicines, new bio-based engineered materials, also production for plant material, and construction. And in the end, this puts in the hotspot Bactris guineensis as a prioritized useful palm species in Colombia's bioeconomy. So in this regard, this was very interesting for me to tackle this question. What are the challenges and the strategies to formalize this value chain? So my purpose during this PhD process is basically to assess the palms value chain in Colombia and to propose strategies that aim to position the species and its derived products as a sustainable and inclusive business opportunity. And to do so, first, I am analyzing the current state of the chain in the country. I am identifying the critical factors that influence its development, and I will propose in a participatory manner strategies aimed at upgrading the value chain under a sustainable rural development perspective. And throughout this process, I am basically using both primary information and secondary information. So one of the main results I've seen during this research when gathering secondary information was that there is a high biomass potential when considering the harvest of Corozo fruit in the Caribbean. When analyzing the trade of this fruit in the biggest city of the Caribbean region, that is Barranquilla, you can see that averagely 15 tons per day are traded, which means that per harvest, there have been traded more than 1,000 tons, and this is unprecedented for a non-timber forest product in wild systems. Secondly, that from secondary sources, it is the first time that we present a picture of what is happening on the value chain in Colombia. And as you can see in this picture, firstly, it's a developed, highly complex value chain, and highly complex, especially in the access to the resource, with diverse interlinkages and relations. And that we are not talking only about raw materials, but we are able to be talking about primary transformations of natural ingredients, specifically for the pulp, but also we're talking about secondary transformations for cosmetics and for food and beverages industries. However, like one of the key results also was that there are some critical challenges we need to work before thinking of strategies. And even though we have a well-developed value chain, it is really characterized by informal relationships. And in the end, what does this mean? It means that providers are failing to meet the quantities that are demanded by clients and their quality standards. And in the end, if we go back to understand this has been due to the lack of long-term agreements and alliances, the lack of robust scientific information or community-based information in monitoring, and the lack of capacity building processes in partnership development. Another key challenge was that the prices of the raw material are very volatile. And in the end, this is explained by the unpredictable yield in wild systems due to the influence also of climate change, the lack of on-storage and processing facilities. And another really important factor that is also affecting the chain is that harvesters do not own the land. So they have conflicts with landowners that are usually livestock rangers and farmers. And for this segment of the chain, it represents an opportunity cost to be using Corozo because they want to expand the agricultural frontier, and this has represented in the end the diminishing or the decrease of Bactris guineensis populations. So for these actors, Bactris guineensis is still not seen as a business opportunity. And if this doesn't happen in the first link of the chain, in the second and third link, what do we see? We see low value added applications, and we see an underutilized potential of byproducts. But I don't want to be negative. Not everything is bad. I must say that there is a regional market that is well consolidated, expanding nationally, and that this fruit has really interesting organoleptic properties that can be compared very similar to acai due to its antioxidant components that are really interesting for niche markets, but also to access to certifications that recognize environmental, social, and health claims of these products. So what are we doing now? We're gonna propose targeted strategies for this value chain development, and we're gonna make a cycle of workshops and focus groups with diverse direct and indirect actors of the chain. And in the first stage, we're gonna ask together, where are we headed? Where are we going? What is the ideal state in which the chain should go in terms of its products, applications, and sectors? And we're gonna try to put this down in a vision of short, medium, and long term. When we have this, then we are gonna ask ourselves, what do we have? With what do we count? What are our capacities and strengths? What are our challenges and barriers? And this is going to be contrasted with the secondary information that I just showed you. In the stage 3, we're going to know which is the ideal state and what do we have. So now, how are we going to do it? What are going to be these concerted actions and agreements? What do we need to accomplish them? Who are the responsibles and who is involved? And finally, this PhD project wants not only to give back information to socialize, but also to serve as a baseline to continue for project proposal for funding with these strategies. And thank you very much for listening to me. I'm really open to questions. Thanks a lot.
Thank you, Tatiana, for a really interesting presentation. I did not know that one of the most important Colombian folk instruments was made out of that plant. To start with questions, I wanted to ask you, do you know how big is the area that is planted with these trees currently in Colombia?
So right now the plant is not planted, is not cultivated, it's in the wild systems. So that's very interesting. Like the Humboldt Institute, that is the National Biodiversity Institute, has all of this data. But what can I tell you is that the populations have been diminished due to the expansion of the agricultural frontier. So that is a really critical issue because a lot of people, their livelihoods depend on this palm.
And I wanted to ask you, will you expand a little bit more on how the conservation program is trying to assess or what are the future plans for conservation programs for this plant?
So basically, right now, this species, like I'm talking from my PhD project, I use the approach of using to conserve, right? I don't talk about like conservation as a world that you cannot touch. So my proposal is that we can preserve the tropical dry forest ecosystem by sustainably using this palm and revalorizing the use of the palm into an economy, but not into an economy, into a socio-biodiversity bioeconomy. So that is my approach to conservation, basically.
Okay. Thank you so much for your presentation. We will continue with our next speaker. If there are any other questions, we will continue with them on our discussion. So our next speaker is Viviana Salinas. So she is a researcher at the Humboldt Institute and she's a forest engineer. And she's a forest engineer of the Francisco José de Calgas with District University in Colombia. She's currently pursuing a master's degree in forest management, use and conservation at the same university. And she has experience that includes long-term monitoring of plant populations, analysis of functional responses to environmental factors, and implementation of ecological restoration projects in collaboration with some local communities and international organizations. So Viviana, over to you.
Thank you, Gina. Okay, hello everyone. I want to start with thank you for the invitation to participate in this space where I will talk about two topics I am very passionate about, the tropical dry forest and the functional ecology. Okay, to start, the environmental factors have been in determining the composition and structure of tropical forest. It has also been demonstrated that environmental variables such as climate, soil, and land cover transformation through different processes such as drought, nutrient limitation, and the introduction of the new species of plants caused by the land cover transformation. These factors, acting individually or together, are referred to as environmental hardness, which can restrict species establishment. These conditions occur especially in the tropical dry forest. The tropical dry forest present great variability in the environmental factors, such as the fluctuation in precipitation and temperature, have highly heterogeneous soils, and have a historical alteration of the vegetation cover. This picture is the tropical dry forest in the Guajira, Colombia, in the Caribbean zone. So to continue, the effects of the environmental factor lead to a decrease in the forest structure and taxonomic diversity. When as drought conditions increase, the nutrients decrease and the land cover transformation intensifies, meaning great environmental hardness, forest structure, and taxonomic diversity decrease. However, the ecological mechanisms behind these changes are still unknown. Therefore, where I will analyze this true functional ecology, which allows to understand species ecological strategies, be evaluate their traits and relating them of the physiological process. Therefore, evaluate the functional response at the community level will allow to understand the variation of the community weight and mean through the optimal trait value within in the community. On the other hand, the functional readiness allows to understand the range of the trait variation under in the environmental limitations. So we ask how good the plant composition and functional diversity in Colombian dry forests vary along a gradients on environmental hardness. For this, the first be establishing 98 plots, permanent plots. We explore the environmental factor, include climate, soil, and land cover transformation. To understand the mechanisms, we quantificate 7 anatomical traits and 3 leaf traits associated with the plants. Once of the material was processed, these take a long time. We've moved on the data analysis phase, where the first step is calculate the indices of community weight and mean and functional readiness. With this, performing a principal component analysis for both environmental factors and functional traits to understand their spatial distribution. Once environmental variables and response rate were standardized, we apply the spatial length model, this spatial regression model that considers spatial dependence. We examined a spatial autocorrelation in detail to obtain the response variables and generate the best model for predicting community weight and mean and the functional weakness. In this sense, our results indicate that they were climate explain the 70% of the variance. In the first axis, we observed at the gradient of the water availability, it's the drought. The negative values is associated with a lower drought, for example, higher annual precipitation, and the positive values reflect drier condition with high temperatures or greater solar radiation. Regarding edaphic variables, they explicate the 52.38% of the variance. The first axis describes a fertility gradient. The positive values indicate soil with high organic material and basis totals and higher water retention. The negative values indicate the sandy soils with lower nutrient availability. For the landscape transformation, explain 62.8% of the variance. The first axis represents a land use gradient. The negative values correspond to the plots with high forest cover, and the positive values indicate greater human interventions. In this sense, our result determined that the environmental hardness in Colombian tropical dry forest is determined for the gradient of the climate, soil, and land cover transformation. To understand how functional composition behaves, our results confirms that the tropical dry forest community follow the trade-off between hydraulic safety and efficiency. When the communities oriented to the safety strategies have like traits, high good density and slower water conditions, while the communities oriented to efficiency strategies have associated with a low mechanical resistance and lower investment in tissues. This pattern confirms previously studies showing that in higher environmentals, when the conservative strategies dominate, reducing embolies and favoring resistant structures. Regarding the functional readiness, our result doesn't decrease uniformly of the extremes of the gradient. Most plots have the similar values of the functional richness, and there has not significant correlation between functional richness and species richness. The absence of the correlation with species richness suggests that the variability functional strategies is not necessarily linked of the number of the species, but rendering the functional distribution within the ecosystems in general. To evaluate the effects environmental hardness on functional compositions, our analysis indicate that the combined climate and soil model explain the 37% of the functional variability. The temperature and the soil variables had a negative effect, while the vapor pressure deficit showed a positive effect. The soil variables influence the dominance of species with conservative strategies. In fertile and transformed soil, these species invest in the restant tissues to resist drought conditions. It's important to know that the land cover variables not significantly affect functional compositions, such as that the functional traits in the tropical forest have low plasticity to this change. We also analyze the factor that modulate variability in functional richness, and we found that the variables of the climate and the land cover transformation respond when the main determinant functional richness were annual precipitation, the positive influence, and the water deficit in dry months, these promoting drought-adapted strategies. The solar radiation is a positive interaction. This is strongly associated with greater functional diversity. And landscape transformation is a negative influence where weather fragmentation leads a lower functional richness. The wind, the negative affects the functional richness by selecting resistant species. For example, the higher wind speed generates mechanisms stress, favoring optimal survival strategies, but reducing functional diversity. Finally, some take-home message is the functional composition of the tropical dry forest respond to the environmental hardness. This is associated or determined for the multiple factors in the variables to study through environmental filtering process. These findings emphasize the importance to understand a functional diversity beyond species continuum. This is key for the establishment and adaptation of tropical dry forests. And finally, environmental hardness determines the functional composition and richness in the tropical dry forest. They need considering multiple factors for the take conservation and management. Finally, thank you for all people who participate in the beautiful process. This is a pictures for the tropical dry forest in Colombia, specifically in the Caribbean, in the Tayrona Natural Park, and the Guajira is the tropical dry forest in the most expression of the drought. Thank you for the Red Bosques Ecotropical de Colombia and Instituto Humboldt. Thank you so much.
Thank you so much, Viviana. It was a really interesting presentation. Do we have any questions from any of the audience or in the chat? Perhaps, let me just check. Okay, so no questions for now, but I can start with the questions. I wanted to ask you, do you take into consideration different aspects like altitude? Or
Yes.
it doesn't influence much?
Can you respond in Spanish?
Sorry.
Don't worry. No. Te puedo responder en español, te entendí, pero te puedo responder en español.
Sí, pues será para traducir, pero dale.
Ok. Sí hemos considerado estudiar la altura en el bosque seco tropical. Es una de las variables que inclusive más se han estudiado. Entonces quisimos explorar justamente las variables que necesitamos entender su respuesta. Gracias.
Ok, Okay, so if I understood correctly, and also to translate for all the other audience, then the altitude is being one of the variables that has been the most studied. Oh, sorry, I just had the alarm. It has been the most studied for the dry forest, and you're taking it into consideration for all these models, right? And then a second question that I had is, for how long did you collect all the data for the climate?
Use the platforms of the climatic in Colombia because it's more establishment in the data.
Okay. Thank you so much. Thank you so much, Viviana. We will continue with our next speaker. So our next speaker is Dr. Kira Lynn. She is the first female she is the first female that was selected as part of a collaboration project between research in academia and industry between FABI and the Royal Golden Eagle. She has experience in Indonesia forestry sector, specifically on research and development, and she also has a lot of experience on diagnostics, developing diagnostic tools, population genetics, and epidemiology. So Kira, over to you.
Thank you so much, Gina, for the introduction, and thank you to the organization committee for the invitation. As Gina suggested, I'm quite passionate about forestry management, and so today I'll be discussing a fungal pathogen and its associated disease known as Ceratocystis wilt, and how fundamental research can help give insights into how to manage this disease. So a bit of background, Ceratocystis wilt affects a number of important agricultural and forestry crops in a number of different regions around the world. The causal agent of this disease has been identified as either one of these several Ceratocystis species that reside in the Latin American clade of the genus, otherwise known as Ceratocystis eucalypticola, Ceratocystis manganecans, and Ceratocystis fimbriata. So despite best management practices and efforts to reduce the impact and spread of this disease and its pathogens, new disease outbreaks have been reported in recent years on new hosts as well as in new regions. So with these new outbreaks that are being reported on these previously unaffected regions as well as on these new hosts, it becomes really important to be able to accurately identify the causal agents of this disease, as well as understand how they're being dispersed to better manage or effectively manage these disease outbreaks. So to go into a bit more background, Ceratocystis fimbriata, although it has been identified in literature as one of the causal agents of this disease, it actually is not associated with this disease at all. It is phylogenetically closely related to Ceratocystis eucalypticola and manganecans, but this species only infects sweet potatoes, and based on population genetics, it's shown to be quite a clonal population that's causing this disease outbreaks. Whereas Ceratocystis eucalypticola and manganecans, which are associated with Ceratocystis wilt, are again phylogenetically closely related species. They're morphologically quite similar, and the symptoms of their disease outbreaks on different hosts is also very similar. But where they differ is they have different host ranges in which they infect, as they present different disease epidemiologies, with Ceratocystis manganecans usually causing infections on the main stems of the host they infect, whereas Eucalypticola follows more of a soil-borne disease progression. So despite these differences, the taxonomic identification of these two species is quite contentious. But because of these differences, it becomes quite important to accurately identify these pathogens in order to effectively manage them. So why is there this taxonomic debate? So as I said previously, Ceratocystis eucalypticola and manganecans are phylogenetically closely related species within the Latin American clade. A single isolate can have multiple ITS types, and as ITS has previously been used as a barcoding gene region to identify species, it has created quite a large amount of confusion in the literature. They are morphologically quite similar, as well as the differences within the gene coding regions used to phylogenetically identify these species. There's quite a limited amount of variation. Furthermore, individuals of both species are found to be able to mate and be interfertile in laboratory conditions. Therefore, depending on the species concept you use to identify species, these different pathogens have either been identified as different species or can be found as conspecific of one another. And this has been quite confusing in literature. It's limited abilities to have comparative studies and hindered effective management strategies of these pathogens. So what's a potential solution? So one potential solution is using a pluralistic approach in which you test the concordance between the species boundaries implied by different species concepts using different data sets. And this is what we did during my PhD, but for the purpose of today's talk, I'll be just touching on two of the biological species concepts and how they can impact effective management. So this will be the phylogenetic species concept and using population genetics. And this will, again, go back to the core of what this purpose was to accurately identify these species, but also gain insights into how they're being dispersed. So for the data set to perform this analysis, we had about 1,192 isolates that were collected across 11 different countries and 8 different hosts. This data set came from historical disease outbreaks as well as new disease outbreaks. So moving on to phylogenetic analysis, we screened this population using 8 gene regions. And what we found was that these 3 gene regions were the most informative in being able to distinguish between Eucalyptus cola and manganecans. And using a maximum likelihood phylogenetic tree, I'm going to demonstrate to you the main findings that we found. So what we found, we were able to accurately identify all of the isolates as either Ceratocystis, Eucalyptus cola, and manganecans, and we found that these 2 species are phylogenetically distinct. The other thing that we found that's quite important is there was evidence of admixture, and I'll touch on to why this is important a bit later. Then moving on to population genetics, when screening these isolates based on their country of origin, regardless of whether they were identified as manganecans or Eucalyptus cola, we find a very similar story to what we found with the phylogenetic analysis. So in this image that you're seeing, each line is an individual, and then the numbers correspond to the country in which that individual is from. So if I can paint the picture for you here, if you look at Indonesia, you'll find individuals that group with manganecans and you'll find individuals that group with Eucalyptus cola, but you'll also find individuals that have evidence of admixture, which is exactly what we found in the phylogenetic analysis. We could look at this another way using a DAP-C analysis, which essentially the story is the same, is that these isolates consistently group into 2 distinct clusters that correspond to the species Eucalyptus cola and manganecans. We can also look at different dynamics using this population data, and one of those is looking at the multi-locus genotypes. So what you're seeing in this figure is these circles are different multi-locus genotypes, and the colors correspond to the countries from which they originate, and the size of the circles indicate how many individuals have that multi-locus genotype. And again, we see that there's this distinct clustering of these isolates into cluster 1 and cluster 2 that correspond to Eucalyptus cola and manganecans. And to demonstrate this, if we look at Brazil, which is colored in blue, all the isolates group with Ceratocystis Eucalyptus cola. Whereas if you look at Indonesia, you have individuals that cluster across the 2 groups. But the other really important thing that this is demonstrating is that if you're doing resistance screening trials, it's really important to know what species you're screening your clones with, and if you're working in plantation forestry. But the other thing that's really important is that showing that there's a high amount of diversity within a species. And it becomes really important to basically screen the diversity of these isolates against your clones to see if you're accurately representing resistance within your breeding strategies. So as I said, going back to the hybridization or the admixture that we found was hybridization was detected in 4 countries, namely Brazil, Colombia, South Africa, and Indonesia. But why does this matter? So hybridization really does complicate our ability to accurately identify these species. And this further has impacts on management because these hybrids could have more pathogenic behaviors, they could have a broader host range, and they could overcome the resistance measures that have been put into place to control this disease outbreak. The other thing that we found in the study, which is quite informative for disease management, is it gave us insights into the different types of dispersal mechanisms we are observing in these two species. So for example, Ceratocystis manganecans seems to predominantly spread through these host expansion events. And this was observed with multilocus genotypes found in both acacia and eucalyptus in both Indonesia and Malaysia. But what we also found is that the spread of Eucalypticola seems to be strongly linked to the global movement of eucalyptus propagation material. And a key indicator of this could be essentially with the new introduction that was found in Colombia, there's shared haplotypes with Brazil, and both of these multilocus genotypes were found on eucalyptus clones. So the other really interesting thing that you find is there's a geographical divide of these two species. And this is important because it informs quarantine strategies. You don't want to be moving propagation material and introducing one species into an area where it doesn't exist, because this can lead to more hybridization events, which can lead to further disease outbreaks and overcoming of your resistant breeding programs. So what's the take-home message or summary of all these findings is that all of the isolates that were both from historical outbreaks as well as newly formed outbreaks or newly reported outbreaks were correctly identified as Ceratocystis manganecans and Eucalypticola. And this helped clarify the literature and gave insights into having comparative studies. It also gave us evidence that there's hybridization between these two species, which confuses identification, but also is quite challenging when you're trying to manage this disease outbreak because your screening material is constantly changing. So why is this important? So of course, accurate identification matters because it's essential for breeding resistant clones and quarantine measures. If you know what you have, you know not to move it. The other thing it informs is preventing spread and managing disease outbreak. It informs quarantine protocols, as I said, but it also, if we monitor genetic diversity, it helps inform resistance breeding again and allows us to prevent further hybridization events. Furthermore, the distinct geographical distribution, host ranges, and the disease progressions of these two species becomes really important in how you quarantine and manage these two diseases. So with that, I hope I've given you a bit of what I did in my PhD and what I'm passionate about. And thank you so much for listening, and any questions are welcome.
Thank you so much, Kira. We have a question in the chat from Pascal Frey. Hi, Kira. In case of admixture of both species, is there co-infection of eucalyptus trees? Does one species outcompete the other?
That's a really good question in which we don't know the answer to. But what I will point out, or what we've noticed, is that, for example, in Indonesia, where a lot of this work was done, is that the manganecans does still seem to be the dominant species. And in compartments where you're finding both species, you'll find more isolates of manganecans than you'll find of Eucalypticola. Whether in a single host there's one outcompeting the other, we're not entirely sure. But there's a lot of research capacity because one of the things we are interested in is one more pathogenic than the other? And I think it also depends on the clones that you have, because as you're seeing, there's differences in the genetic diversities. But that's a question I'd like to answer.
There's a following question from Josephine, and she asked, is it known how long ago these two species diverged?
There's no, as far as I know, there's no specific study into when divergence occurred, but based on the literature that is available, there is evidence that they're newly diverged species. And we believe that they're actually in the early stages of speciation. When the exact time that happened, we don't know. And the problem is because they're still able to hybridize, it confuses understanding that. So yeah.
All right, thank you so much. So with that, we will start our discussion. There is another question from Pascal Frey. So he asked, "Hello, Jane. In case of severe fire, how long can Armillaria survive, and is it a threat for recolonizing tree seedlings?"
So we only sampled 15 months post fire. However, I would imagine that armillaria, once it survived through the fire, is able to survive long term because there's going to be weakened trees on the site. And then when we go in to replant, for sure, armillaria will be a risk for those new seedlings that have been planted. And a lot of times they don't plant, they try to plant when rainfall will happen. But in some cases, those seedlings get stressed after they get outplanted. And so that can facilitate armillaria attacking those trees. Thank you for the question.
Thank you. So we have a question online. So Josephine.
Hi, thanks, Gina. This is for Kira. So this is a follow-up to my question. I'm thinking like if they diverge very recently, I'm thinking that in some areas of the world it's going to be advantageous to keep hybridizing, and so the speciation process is going to take more time than in areas of the world where it would be more advantageous to separate into different species. Is there a protocol for when you have two different answers depending on geography?
So you're saying how to manage them when you've got both species in one?
Well, when you have two different answers depending on where you are in the world, your question of are they two different species? Some people might have different answers because they are studying different populations.
So based on, so we did, so to go to the base question, we did every single species concept that you can do. And based on that, they are distinct species. And when, even if you're working on what you're working on in one country, if you use the biological species concept to describe them, they are separate species. So it's not going to change the identification. Where the confusion comes in is if there's hybridization, because then it's not a clear manganecans or Eucalypticola, it's a hybrid. So in terms of management, if you have one species in your region, it's not a misclassification. You can clearly define what it is, if that's answering your question. Yeah.
Are there more questions from the audience? Please raise your hand so we can grant you access, or if you want to write them on the chat box. Okay, it's a silence audience. So if there are no more questions, I hope there's another question. It says, "Hi, Kira. I'm interested in the different population genetic grouping methods like phylogenetics, admixture, and the APC. They all pointed in the same direction?"
Yes, so I can give you. I did a whole talk in which I went through every step of every species concept and found strong evidence that they're separate. But every single analysis, everything we threw at this, constantly grouped everything into two distinct clusters, regardless. Particularly if you look at the population genetic analysis, we didn't form or group our isolates based on what species they were. We inputted them based on country of origin and regardless of what species they were, and they constantly separated into the two clusters and correctly. So if you have individuals, for example, in Indonesia, if I showed in that figure, you have individuals that are Eucalypticola and you individuals that are manganecans, and those individuals are dividing across in all of the analysis we used. If that's answering your question.
Yes. It says, "Thank you." I can't remember, Kira, but from your work, did you also find both species in the same areas?
Yes, so I actually have just done another population study, and we're finding in the same compartments, we're finding both species and then evidence of hybrids. And something we're really interested in is to kind of look at the genomes and understand how that's happening. But yeah, we're finding a lot of hybrids. If they're in the same area, they hybridize.
Right. Any last burning question from the audience? Right. So without further ado, I hope these sessions has invited you to dialogue, to have discussions, and to have conversations to reflect about the role for women and in also indigenous communities and all of this in science. It's really important to know that the female representation and all the contributions differs depending on the global region, but they all contribute to the same goal, which is to inspire and encourage future generations. So I really hope that you keep on supporting us and that you join us for the next webinar next year. Thank you so much.